Hello everyone. My name is Robert Driver. I'm a postdoc at Duke University. And if it's your first time or if you're returning, welcome to the AOS Student Research Grant Seminar Series. Just a quick intro, the AOS offers students and early career researchers the AOS Student Research Grants each year. And our seminar series here celebrates the challenging and innovative work of the grant recipients by sharing this AOS-funded or related work with the AOS members. So each week we showcase the research plans of 2024, 2023, or 2022 AOS Student Research Grant Award winners in a seminar-style format. We have one seminar each week for 15 weeks. I think we're at the midway point now, going until December 15th. So thank you everyone for being here. The AOS and AOS Student Affairs Committee, Chris Anhandel, Ramona Fletcher, and especially thank Holland, our speaker today. So before we turn our attention to Holland's research, I just want to mention if you have questions, we have a Q&A session at the end of the talk. And you can participate by unmuting yourself. I'm sorry, I say that every time. I gotta change that. By raising your hand using the raising hand icon, and then I can unmute you. And actually, we had a few people do that last week. It was very nice to hear people's voices. But if not, you can also enter your question in the chat box or in the Q&A box, and I'll monitor all these different ways of asking the question to make sure everybody gets their thoughts in. And I want to remind everyone this is an AOS sponsored event and that all participants are required to follow the AOS Virtual Meeting Event Code of Professional Conduct, and I'll post a link to that in the chat. I will also post a link to a post-seminar evaluation form that allows you to provide feedback to help us improve the seminar series as we go week to week here, as well as a contact email for the AOS Student Affairs Committee. And please be aware that we're recording this talk. Okay, so today, Holland Galante is a Ph.D. candidate in zoology at North Dakota State University. And Holland's academic career began with a Bachelor's of Science in Biology at the University of Wisconsin-Milwaukee. And her undergraduate research studied gray tree frog response to visual stimuli and was published in the Journal of Experimental Biology. And following graduation, Holland worked as a research technician in the Medical College of Wisconsin, working on the molecular biology of neurodegenerative diseases. And her research from this technician position is published in Nature Communications. Holland then worked as a water chemist at Milwaukee Water Works, testing for water quality parameters. And then in 2018, Holland joined the laboratory of Dr. Timothy Greaves as a PhD student, studying the mechanisms that initiate the seasonal breeding biology of birds, including fluctuations in steroid hormones. And Holland's research from her time in graduate school has led to publications in general comparative endocrinology and behavioral ecology, among others. And in addition to the AOS Student Research Grant, Holland won the Sigma Xi Research Grant. And in 2020, Holland received honorable mention for the NSF GRFP, which is, of course, very competitive. And with that, I will turn it over to Holland for a talk entitled Telomeres, mitochondrial copy number, organismal metabolic rate, and growth rate. What is the connection in the developing house sparrow?
All right, thank you for that wonderful introduction. I hope everyone can hear me. So again, as Dr. Driver said, I'll be looking at telomeres, how mitochondria is affected as well with growth and metabolic rate and growth rate in house sparrows. So we know that, well, first I'd like to thank a whole bunch of people that helped with this work. We have a couple of previous postdocs who were super valuable to our lab and helped greatly in the study, either with analysis or gathering. Without them, this couldn't have been done. I would like to especially thank Dr. Kendrick Greenlee, who helped us with doing the metabolic rate and setting up the chambers and respirometry for that. So she was essential in gathering that data. As well as I'd like to thank the NDSU Bird Lab, who was very helpful in their feedback and very supportive. Our lab is photoed at the bottom here. We have a great community here. Without them, a lot of this work is not possible. And also the birds and the lovely field cats at our site, which were always fun to play with. Additionally, I would really like to thank AOS for awarding me the winner and Hildegard Hesse research grant. Again, without this funding, I was not able to do this work as well. All right, so birds undergo a lot of different life history traits. So birds can undergo reproduction and also undergo migration at different times of the year, and they are optimized at these times of years for the best energy purposes. So for reproduction, a lot of long day breeding birds. So when I mean long day, they have their reproduction during spring and summer. They often, make a nest and they lay their eggs. So we have two different types of development. So we have eggs during incubation, and then also during this time, we have extreme amount of growth that occurs during the actual nestling growth phase. Here you can actually see on the left, the day 2 chick, just super tiny. And then on the day 10, they are about ready to fledge and they're almost the same size of adults. So this period is such a crazy amount of growth and different changes in physiology are happening. And we want to understand what is actually going on with this. So we know life is expensive in terms of development. And what are the mechanisms that underlie energy demands for growth and longevity? Because those are often tied together. So metabolism is one of the mechanisms that underlies growth and development. So a study done in rodents has shown that you had higher levels of basal metabolic rate. Again, I'll get to it in a couple of slides. There's several different measures for metabolism, but basal metabolic rate is one of them. You actually had shorter gestational periods. So what that means is that you're growing faster in utero or within potentially an egg, in the case of birds, that you're going to hatch faster or be birthed faster. And then we also see this as well as outside of the uterus or in vitro or in vivo. We see that as your metabolic rate increases, you also have an increased growth rate as well. So that development time outside of your maternal conditions. Additionally, metabolism is one of the mechanics that underlies longevity. And a cross study of a meta-analysis of mammals and birds across several taxons, we see that the general trend is a negative correlation where basal metabolic rate actually, as you get higher, you have a reduced lifespan, which is very interesting. So if you have, it's kind of the live fast, die fast ideology with this. However, within each species, and this is a different type of metabolic measurement, it's peak metabolic. Again, I'll get into another slide. You can just think of it as metabolic rate right now, is that if you have higher levels of metabolic rate, you actually have longer lifespans within a species. So we kind of see controversy between these two ideologies of where you have higher metabolism across species having reduced lifespan, but then within a species, you can have higher metabolic rate and longer lifespans. And that was shown in the Gavin-Ville Fitrelli butterfly. So metabolic rate is very complex, and there's a lot of factors that actually go into this. So there's temperature, body size, and rates of other biological processes. So we can think of as body size as affecting metabolic rate. We know that body size and metabolic rate scale together. So if you're a larger animal within the species, you will have usually a larger metabolic rate. It might not be a perfectly 1-to-1 ratio. It's like raised to the 3/4 is how it's scaled, but it still scales with body size. Other biological processes affect metabolic rate. We can think of reproduction as a different life history trait. We can think of migration as one. We change our differences in metabolic rate, where, for instance, birds will actually kind of liquefy their organs for migration to account for that energy expenditure and energy need. Development is another one, which we'll be talking about. And so those will also kind of be all interconnected. And the next, temperature, we can think about on several different levels. We can think of it as the subcellular level with kinetics, where we know that enzymatic activities increase with temperature, so that can increase your metabolic rate. We can also think about it as a thermoregulation needs as well, where we see changes in temperature, you need different thermoregulation needs, where if you're a colder animal or in a colder environment, you need more warmth and heat so you can up your metabolic rate in terms. How body size and temperature are connected is that we know temperature can increase growth, especially during development. Also, interestingly, too, we can see that during, if we look across a latitudinal gradient, we see birds in the north with larger body sizes and birds in the south that have smaller body sizes. So, temperature is affecting how body size is related to their actual geographic position, which then can also affect their metabolic rate. Again, going back to the body size scaling factors, as well as temperature as well. Because we know northern birds can experience more extreme cold temperatures, whereas in the south, they don't. So, all right, so kind of taking a step back. So, where does our energy come from? Going back to basic biology. So, we have aerobic respiration. So, that is the breakdown of glucose with oxygen to form carbon dioxide with water and energy as ATP. So, where does this exactly occur? We actually have some occurring glycolysis that occurs in the cytoplasm of the cell, but the vast majority of the energy produced is in the citric acid cycle as well as in the electron transport chain. I should say more of it is actually produced by the electron transport chain in the mitochondria. So the mitochondria plays a vital role, and we always think about the mitochondria as a powerhouse of the cell. Like in biology, that's the running joke, but the mitochondria really is a powerhouse of the cell, and it actually has a lot of factors that affect it. So how do we measure metabolism, or what is it, or metabolic rates? It's the amount of energy expended by an animal over a certain period of time. And there's a couple of different ways we can measure it. Again, as you saw in the previous slides, it showed basal metabolic rate and peak metabolic rate in those studies that I presented. So the first one is forced resting metabolic rate is what we'll be focusing on in our study specifically. That's why I've highlighted it in red. So that is when you take an animal during its awake phase and then force it to rest in a container to measure the metabolic rate. So it's basically kind of making it stay still. It's not really an actual state of sleep or non-activity because they are still awake. Summit and peak metabolic rate is actually measuring the height or the amount, sorry, I should say, measuring the maximum amount of energy an animal produces during exercise or physical exertion. It's kind of funny to watch this because in birds, they have like a ping pong machine to keep the birds in flight to get them to keep in motion and keep exercising. So that's another form of it. And then basal metabolic rate is the minimum energy of an animal required to live at a post-absorptive rest or post-absorptive phase in their resting phase of their cycle, their circadian cycle. So again, we will be looking specifically at forced resting metabolic rate. And so how do we measure this? So we specifically use a flow-through respirometry where we put a bird in a chamber and we have a known amount of oxygen coming in to the bird's chamber. And then as the bird consumes oxygen and the air exits, we can measure the difference between the two to get the metabolic rate. So let's go back to the mitochondria. So again, I was saying the mitochondria is very important because it produces the vast majority of our energy. And so changes in mitochondria abundance and function can influence your metabolic rate, your energetics. You can think of thermoregulation and cellular respiration, as well as biological processes, i.e. growth, and also migration and reproduction. And so we can measure these as function is measured by the actual cellular respiration of the mitochondria. We can actually measure how much oxidation phosphorylation is going on at each different stage of the electron transport chain. Or abundance is measured by mitochondrial DNA copy number. And that's what we're going to be focusing on today. So specifically, again, we'll be looking at the amount of mitochondrial DNA, which is a proxy for the density or how much mitochondria that you have in a cell. So what we're looking at is that mitochondrial DNA copy number is actually a ratio between nuclear DNA and mitochondrial DNA. So when we think of nuclear DNA, it's our chromosomes within our nucleus and usually only have like a single copy, whereas mitochondria has several copies within its own every single mitochondria. And so just here pictured is just the general genome of what a mitochondria genome looks like. We have 22 tRNA encoding proteins, our genes, and then 13 protein encoding regions. And so this is highly conserved across most taxons, where you see the circular DNA and that is kind of in the same order. Some genes might be changed a little bit in it, but it encodes for all of your electron transport genes. And specifically, we'll be looking at the gene cytochrome B, which is near the top in this darker green color. All right, so continuing on what mitochondrial copy number actually is. So each mitochondria has between 2 to 10 copies of DNA within them. Again, the mitochondria kind of hangs out in the matrix of the mitochondria between the cristae within it. So that's kind of where it hangs out. And then each cell has a lot of mitochondria, right? So then that can range, the actual number of mitochondria DNA can range from 10 to the third to 10 to the fourth. So that's a crazy amount of range from different cell types. And again, different cell types have different energetic needs, and so they'll have variations in this as well. Mitochondria, the number and the function can be modulated by hormones during different life history stages or under stressful conditions. So we have seen in several studies, one was done in the brown trout, during starvation periods, actually mitochondrial DNA amount decreased. So hopefully preserving those energy demands for other uses other than, if we want to preserve our function and just general living and not go into reproduction, then we need to decrease our energy demands. I also have seen within life history traits in the pied flycatcher, actually we see the amount of mitochondria and the function of the mitochondria actually changes from incubation to chickering. So that is actually saying, hey, we need completely different energy demands during these different reproductive stages, and that's potentially linked to incubation and heat production. All right, so the amount of mitochondrial DNA is actually proportional to the energetic demands. And that's what I mean, it is tissue-based. So when we have lower metabolic tissues with lower turnover, so lower turnover means how often these cells are dividing and replacing each other, we'll have a lower mitochondrial DNA count. Vice versa, with higher metabolic tissues, we'll have higher turnover rates and have higher mitochondrial copy number because they have higher metabolic demands for energy. All right, so again, we're specifically going to be looking at blood mitochondria. It is easily accessible and is easily stored for later use. When we look at mitochondrial function, a lot of studies like to look at the actual respiration rate, and the vast majority of them look at this function of mitochondria with 24 hours. So that might not be feasible for some people if they cannot get to a lab to actually run these. So we were trying to think of an idea of like, okay, we can take a blood sample, which has been shown to be proportional to energy use, and we can potentially do it later when we have time after the field, weeks or months later, because DNA is stable. All right, and also we want to use blood mitochondria potentially to replace metabolic measurements, which can be stressful in organisms. Even though we try to mitigate how stressful conditions are in organisms to get actual metabolic accurate measurements, there's some kind of stress with either being in handling stress or there's stress from the container itself. It's just not their natural condition. So it could just be a potential way of, okay, we can just take a quick blood sample. It's a quick snapshot of this organism's metabolic rate, and we can just go on our merry way with that. Also, the idea of using our blood mitochondrial copy number is to replace terminal sampling potentially. You can look at different tissues like liver and muscle that are also metabolically active during these time frames, but that usually requires a terminal sampling. And so if you want to monitor how metabolic rate changes across an organism, especially in field environments, blood might be hopefully it would be an answer to those questions. Again, blood mitochondrial function has been shown to be moderately correlated with other tissue mitochondrial functions like liver and muscle, again, making it hopefully an ideal measurement for potentially more field techniques and reducing stress. And so particularly at Casa Grande at 2023 actually showed this. This is particularly looking at the respiratory rate of the actual mitochondria in different tissues comparatively to a whole organismal metabolic rate during the day and night. And so you can see we have red blood cells, brain, liver, heart, and pectoral muscle. And the mitochondrial respiratory rate was actually more correlated in red blood cells for with your organismal metabolic rate both at night and during the day. So again, that's why we're kind of picking blood as this idea of hopefully representing a whole organismal metabolic rate. Again, like I say, the study, this was looking at the respiratory rate of the mitochondria, not the copy number. All right, so continuing on, again, we have some evidence that the amount of DNA of the mitochondria is correlated with several different factors. So first off, I like to say in humans, in lymphoblast cells, this is in vitro, the relative amount of mitochondrial DNA level is actually proportional to the respiratory rate of the mitochondria itself. Within your Drosophila, that mitochondria copy number is positive related with female development time. Again, trying to get at that. How is your copy number or your mitochondria changing with these different life history traits? Like this would be development, which is what we're interested in. Additionally, you can think of stressful events. So this is done in rabbits where they stimulated in vivo their muscle tissue, their skeletal muscle tissues to see how mitochondrial traits changed. Again, this is kind of mimicking stress or exercise. And so we see a positive correlation with the number of days that they were given this stimulus to mimic exercise and stress with citrus synthase, which is another metabolic enzyme that we can look at, which is more on the, it's an assay, it's an enzymatic assay, and it's in the Krebs cycle that it's part of. So it's another measure of metabolic rate. Also, we had an increase in the transcription of mitochondrial DNA, as well as the increase of mitochondrial DNA itself. Again, so there's something going on with exercise and stress that we want to figure out. And then this leads me to now what telomeres and mitochondria copy number might have in common, or how they may be related, is that in humans using whole blood, we see that telomere length is actually positively associated with mitochondria, amount of mitochondria. So that leads me into what are telomeres. So telomeres may be important mechanism for biomarkers of aging. So what are telomeres? So telomeres are repeated sequences of DNA at the ends of chromosomes. They form a cap, a protective cap. So in this picture, you see our chromosomes are blue and our caps, our telomeres, are yellow. So the whole point of that is to protect the DNA through cell division during mitosis, because every time we have cell division, they get shorter and shorter and shorter. And again, that is to protect the actual coding DNA in the chromosomes from having damage and causing mutations. However, since there is a limit to how many times you can cell divide and then finally lose your telomeres again, they get shorter, that limits your cellular lifespan. So your cells will then no longer be able to propagate, and that will lead to aging, as we can see in a lot of diseases. All right, and so since that limits the cellular lifespan, it is used as a proxy for aging in a lot of organ studies to actually have a biomarker of your actual biological age. All right, so individuals with longer telomeres during early life live longer, and we have seen this in several bird species. And here, again, Dr. Britt Heidinger, who I work with, published a study in 2012 where she found that telomere length at day 25 post-hatch, these chicks, or so these birds, have gone through the development phase, and now they're in their adult physiology, but they're still in their early life portion of their longevity. So they found that it was positive correlated their telomere length with their lifespan. All right, so we know that there's a correlation between how long you live and your telomere length. So individuals exposed to stress hormones during early life, like development, had reduced longevity and reduced telomeres. So again, in zebra finches that were actually exposed to early court during their life, and that would probably be done in their nestling phase, they actually had a reduced survival rate. So they died earlier than their counterparts who just received the control treatment. And then also in European shags, we see these nestlings were grouped into unhandled, handled with an oil treatment, which was a control treatment, and handled and then had a quart treatment. So they were actually fed these treatments of oil or quart during this nestling phase. So both in the handled quart and handled oil treatment, we see an increase in quart, and quart is actually a stress hormone. It's a glucocorticoid. and humans experience this as well. So we saw an increase in those two treatment groups, and then we actually had a reduction in telomeres in those two groups as well. So this is kind of saying, hey, stress in general is causing our telomeres to reduce. Again, the handled oil treatment was supposedly used as a handled control to see if handling or court was causing stress to increase in telomeres. But in both cases, handling is also a very stressful thing for these nestlings to experience. So stress can come in many forms, like parental neglect or different environmental factors. And so again, going back to that web, we're thinking about how many different things might influence metabolic rate. Continuing on how stressful conditions in early life cause your telomeres to shorten, it also can increase metabolic rate. So like I just explained the European shake study where we had oil or both handled treatments with quart and our handled controlled treatment experienced a decrease in telomeres. We also see as we have higher metabolic rates, we usually have a higher basal quart concentration. So we have some kind of higher level stress with that is associated with metabolic rate. This was done in a meta-analysis in mammals, I would like to point out. However, how are telomeres and metabolic rate connected? So we're seeing that both ways that stress is influencing some kind of physiological response, but are they actually connected? So that's kind of what we're interested in. So there are very few studies have investigated the link between the mitochondrial copy number amount and metabolic rates and the impacts on growth rate and telomeres across development. So what we're thinking of, or what we're kind of looking at this mechanistic scheme, is that we have mitochondria, which represents our mitochondrial density and how much ATP we're producing. And does that actually relate to how our metabolic rate is functioning, and whether that's affecting growth rate and whether if it's affecting telomeres, and if there's a connection between telomeres and growth rate as well. So what are we asking? So we have a couple questions. So does mitochondrial copy number change across development? Does different environmental conditions influence growth rate and whole organismal metabolic rate during this time period of development? Is blood mitochondrial copy number representative of whole organismal metabolic rate, growth rate, again, during development? Is whole organismal metabolic rate related with growth rate across development? And how does metabolic rate and growth rate affect the change in telomere length across development? So we have 5 different questions that we're asking during this study. So this is what our predictive outcomes are. So we predicted that if we had a higher number of copy number, meaning that we had a higher density of mitochondria, we're going to be producing more energy. So we're going to have a higher metabolic rate. And then since we have higher amount of energy, we're going to have a faster growth rate. And then faster growth rate will actually cause a greater change in telomere length. So when we mean change in telomere length is whether they're shortening or lengthening during this time period. And so we would predict that actually if you have a higher growth rate, that more turnover of cells as you're rapidly developing, you're actually going to shorten your telomeres length because you are experiencing a much faster rate of development. Whereas if you had lower levels of mitochondrial copy number, you would have theoretically lower mitochondria and lower ATP, and so lower metabolic rate and lower growth rate, and a less change in the telomere length. Since you have a slower rate of development, you might not be experiencing that much telomere loss during this time frame. So kind of going back to this time or this web of how metabolic rate is kind of interacting, what we're doing here is in order to tease apart how different aspects of development and stress might be affecting metabolic rate and telomeres, we're manipulating some various factors. So what we're going to do is we're going to manipulate temperature through nest temperature. And then also we're going to manipulate other biological processes as feeding rate, because that is your energy supply or demand during chick rearing, is how well are your parents feeding you. All right, so I would like to introduce you guys to our field site and our lovely species, the house sparrow. Our field site is a very rural field site. It's at a farm. We have a sheep farm and a dairy farm as well. And I mean, it's far in North Dakota, so there's lots of farm fields around. But a lot of people think of house sparrows as more of an urban bird because they are so opportunistic and have cohabited with humans for a while. So that's why I want to make a very big distinction that these birds are more rural birds. And also here in this population, they have little nest boxes that they use during the breeding season. You can actually see a female sticking her head out up in here. Maybe not very well, but she's in there. And also, since we live in far North Dakota, we can have very crazy variations in weather where we can have snowstorms at the end of May. This happened quite a bit actually during the field season. And then towards the end in September and August, we can have over 100 degrees. So there's a lot of variation that could greatly impact how these chicks are developing and how their metabolic rates are responding to these factors as well. So in order to test this, again, I said we're manipulating temperature and parental care. So the house sparrow usually, their clutch consists of 5 eggs. And after the 5th egg, the next day or later in that day, they will start incubation. And day 0 of incubation is when we denote when they started incubating after clutch completion. And then that typical incubation lasts about 12 to 13 days in this house sparrow population. So from day 0 of incubation to day 8 incubation, chick or eggs were removed from the nest for 3 hours a day, every day to be cooled, or they were a control group. So when the eggs were removed, they were actually replaced with like the polymer clay eggs or fake eggs that they'll sit on. They can't tell the difference, so that's why they didn't abandon nest. So it's kind of funny because they just don't know the difference. But so that was mimicking temperature stress on these birds through parental neglect of how well these eggs are being incubated, and we can see differences in behavior and incubation as well in development for chicks or nestlings, I should say. Once that phase was done, hatch day was day 0. And then on day 2, day 4, day 6, day 8, day 10, we had morphological measurements taken, including mass, and we calculated the growth rate from the slope of the mass across those days. And then on day 2 and day 10, we took a blood sample to look at mitochondrial copy number and telomere and how it changed across that time period. And then also on day 6, we looked at metabolic rate. And then during this time, like I said, we're mimicking some other stressful conditions and parental feeding and parental neglect. We had control chicks, and then we had nestlings, and we had stress nestlings that were taken for the nest an hour a day. So in total, each nestling could belong to one of these 4 treatments. So you could have been a controlled temperature chick or nestling. You could have been a controlled nestling for stress. You could have been a cooled nestling or egg and a controlled nestling for stress, or a controlled cooled egg and a stress nestling, or a cooled egg and a stress nestling. So there's 4 different treatments that they could have fallen in because we were taking the same eggs prior to our stress chick treatments. And then we did do some interactions across these treatments to test statistically for that. All right, so what we again were looking at was our forced resting metabolic rate in these chicks, because again, we were taking them during the day, so it's not technically resting metabolic or basal metabolic rate, which has to be done during their nocturnal phase and of course absorptive phase. So nestlings were removed from the field and masked, and then they were brought to the lab, and we have these like little jars, they're little mason jars. They're kind of funny to see them in these mason jars. They're hooked up to all these tubes. I would let them acclimate to this jar for 30 minutes. Then in a separate jar that was empty, we ran a 5-minute baseline to get the amount of oxygen in our air source. And then we switched to the chamber with the chick and did a 10-minute measurement. And then after that, we switched back to the control to get another baseline. And we repeated this for each nestling. So nestlings only received 1 measurement over this time frame, but again, depending on how many nestlings we had each day, we repeated this. Birds were then returned to the field and masked again. Again, I want to note that the float rate was 1,000 milliliters a minute, and the temperature was held at 30 degrees Celsius, which is within the thermoneutral range of house sparrows, at least in northern environments. So oxygen consumed was averaged over the 10 minutes and then subtracted from the baseline and then corrected for the flow rate. And then the forced resting metabolic rate was mass corrected. All right, so our laboratory technique. So from red blood cells, we extracted our DNA. We did qPCR, And then we ran telomeres and our copy number, which is our cytochrome B gene that we're specifically looking at. So interestingly, we found that on day 2 and day 10, mitochondrial copy number are actually positively correlated. So meaning if you are starting higher amount of copy number or density of mitochondrial DNA, you actually will have on day 2, you'll have the higher amount of mitochondrial copy number on day 10, which I think is very interesting because this really I haven't seen much of this in the literature, which is why I picked this study. Also, we see that day 2 mitochondrial copy number decreases across year, but not in day 10. Even though they are correlated, there is some differentiation of what is happening between these different time periods within development, and especially with a year interaction. Again, so day 2 could have some kind of global environmental effects, as like we have weather conditions or differences in food sources. You could maybe have more higher nutritious food sources later on, so you don't have to work as hard to get as much energy. Again, we have higher temperatures later in the year, which could influence that specifically at day 2. Also, another really interesting thing that happened we saw is that mitochondrial copy number decreases from day 2 to day 10 across almost all of our chicks. This is significantly significant. And so this could be from some kind of pruning process we can think of, that in the beginning of the development, we have to have a huge amount of energy. And so we have a lot of mitochondria. But as we get closer to day 10, we have less mitochondria and we're getting close to our adult phenotype. So we don't need as that much energy since we've already passed that huge high energy demand point during development. And I would like to say that most of the growth and development that is rapidly occurs from like day 2 to day 10, and then it kind of has a little asymptote where you slowly level out with how fast you're developing. And so that could be why we're seeing this huge decrease from day 2 to day 10. We also see this process in a lot of other, or we have seen this before in a lot of other studies where, for instance, in human development and neurodevelopment, we see it undergoing a pruning of dendrite densities before birth or development. And also in pregnant women, we also see a decrease in white matter and gray matter. So this might be some kind of pruning process to help streamline our energetic needs later on in life. All right, so let's look at factors that impacted metabolic rates. Again, we're looking at forced resting metabolic rate. We looked at day 2 copy number, day 10 copy number. We looked at hatch date, since we see that that has seemed to have some influence on the mitochondria, especially at day 2. Looking at nesting treatment and chick treatment as well. Again, nest treatment was cooled versus controlled during eggs. And then our nestling treatment was stress versus control during their nestling phase when they're growing. We did not find anything with our copy number, hatch date, but we did find something with our nest treatment and our chick treatment, which is kind of interesting. So both nest and chick treatment increased our forced resting metabolic rate. But again, there's no significant interaction between treatments. Again, going back to the 4 groups you could have fallen into as a nestling, there was no interaction between if you were a cooled, or controlled, controlled stress, or cooled, controller, cool stress bird. So there's no interaction there. But we do see that, hey, this is potentially a stressful condition. Being cooled as an egg, right? So that might have some kind of effect. And also being removed from the nest is stress, whether it be limiting on food or stress from handling. We see that it's increasing, and we see this is kind of mirroring results that were previously indicated, that where you have higher rates of metabolic rate had higher rates of cord level. So we're kind of seeing similar results here. So does mitochondrial copy number change across development, for our first question? It does, again. So day 2 mitochondria decreases across the year, and then it also decreases from day 2 to day 10. Does different environmental conditions influence growth rate and metabolic rate during this time? It does. So we see cooling eggs increase resting metabolic rate, and our stressing also increased resting metabolic rate, again, mimicking very stressful conditions as well. All right, so now let's look at factors that impact growth rates. So again, we looked at our copy number for day 2 and day 10, hatch date. We looked at metabolic rate, a nest treatment, and chick treatment. And we didn't really find anything, which is very interesting itself, since we do see some kind of changes related to stress and also timing within physiological changes of the mitochondria. So our next two questions is blood mitochondria copy number represented in full organismal metabolic rate and growth rate across development? The today fact, no, at least in our study, is whole organismal metabolic rate related with growth rate across development? Again, that's no. All right, so finally getting to telomeres. So factors that impact the changes in telomere length from day 2 to day 10. So again, we're calculating this as we're having our day 2 telomere length subtracted by our day 10 telomere length minus our day 2 telomere length to calculate the change across development. So again, we look at the copy number for day 2 and day 10, hatch date, metabolic rate, nest treatment, and chick treatment. So everything but hatch date did not have an effect. So hatch date did, which is kind of very interesting itself. We're seeing this date effect again in our telomeres as well as our mitochondrial copy number. So what we found is that the change in telomere length increased over the year with hatch days. So if you were hatched early in the year in May, you potentially had shorter telomeres on your day 10 relative to your starting telomeres again. So this is all relative to the individual. So it means somehow your telomeres are shortening during that process when you're developing early in the year. Whereas if you're later in the year in August, when it's warmer, you might have longer telomeres on day 10, again, relative to your starting telomeres on day 2. Again, just for reference, we have another day 2 and day chick pictured here to actually kind of emphasize how crazy this development phase is happening. So there could be many causes for why we might be seeing this. It could be the global nest environment, like we see variations in nest thickness, so maybe there's something going on there. I know we were mimicking stressful conditions with removing the eggs and removing the chicks, but maybe there was some kind of compensation the female was able to do over the season, or maybe the nest construction itself, or something physiologically with the female is maybe able to transfer heat better to compensate for those changes as well. Again, I was saying, again, Fargo is very variable, so it could be a temperature of actual environmental temperature influence. Or it could be some kind of food influence too, that maybe you're getting much more nutritious food during later in the seasons. We commonly associate, at least at our field season, that grasshoppers are the preferential food source for these house sparrows, but they don't usually emerge till later in seasons during the warmer summers. We're actually having another PhD student actually look at eDNA through fecal matter to actually kind of characterize what is actually being fed across the year. And so maybe that might have some kind of effect on telomeres as well. Additionally, I want to note that telomeres during this development time actually have an enzyme called telomerase that is highly active, and it's to help, we have this rapid division going on, but it's also to help prevent too much telomere loss. So it's very active during development, where it's actually extending telomeres during this time. And that could be temperature related, right? So we think of enzymatically that it could be potentially more active at this time. All right, so how does whole organismal metabolic rate and growth rate affect change in telomere length across development? Well, it really didn't. We saw a hatch date effect where it increases the change in telomere length. So going back to our predictive outcomes, we didn't really see what we thought we were going to see. We did see some very interesting other date-related effects. And then going back to our predictive mechanism, again, we didn't see what we thought we would have predicted to see for how copy number and density, which could influence energy production and how it could affect your metabolic rate and growth rate in telomeres. So kind of going back again to this scheme when we're looking at how we are manipulating these factors to see how metabolic changes, we found that nest temperature did affect it and feeding rate potentially affected by stressing the chicks by removing them from the nest. So we did see some kind of interaction with that metabolic rate. However, the underlying mechanism still remains unknown. So going back for a quick summary, just to kind of get this in your head a little bit more, is mitochondrial copy number from day 2 decreased across the year. And then also decreases from day to day 10, potentially through maybe some kind of pruning process, getting you towards more your adult phenotype. There was no relationship with growth rate and metabolic rate. For metabolic rate, both cooling the eggs and stressing the nestlings increased it, again, mirroring or kind of reflecting previous studies about stress and metabolic rate. No relationship with telomere loss and growth rate. And for telomeres, we had an increased change in length with hatch age across the year. So for future directions, when I was thinking about this and running these stats and stuff, I was like, well, we have this huge change in day 2 and day 10 mitochondria. So how does that relate actually to our resting metabolic rate and growth rate? So before we publish this paper, I probably will go and investigate this change to actually see if maybe there is it's more related to the change and maybe not on the day 2 and day 10 specifically itself. I would want to investigate the actual cellular respiration of red blood cells and other metabolically active tissues. So again, the caveat with mitochondrial copy number is that, especially in the blood, we don't know what actually percentage or what it's actually meaning to have, what it's meaning for your overall metabolic rate. So we have to do a little bit more research of like, what does it actually mean? So cellular respiration, again, we had a very high positive correlation that we saw with red blood cells and metabolic rates. So maybe that's the way to go, as well as pairing together how the copy number is related to that. And also testing other metabolically active tissues as muscle and liver during this time. Again, such a huge energy demanding part of life during this development phase. Again, that does take away from the potential of not using non-lethal sampling, but it's something we could investigate. Also, one caveat to our study was that we were not able to take blood samples on day 6, the same day as metabolic rate, just the limitation of how much blood we could take from these birds. And so I would love to repeat this experiment, except make sure we take a blood sample on the same day as metabolic measurements, because now we see that we have such a huge drastic change from day 2 to day 10 that we actually might be missing that relationship because we do not have that blood sample on day 6 as well. And also, investing in the health of the mitochondria is important. So mitochondria is susceptible, the DNA at least, is susceptible to oxidative damage because of the electron transport chain. It does produce a lot of free radicals, and that does damage the DNA within the mitochondria. And the mitochondria DNA does not have any real repair enzymes, so it kind of just accumulates this damage within it. So we can assess this with long PCR. And so it actually amplifies the whole genome, the 16,000 base pair genome of your mitochondria. And then you can use a fluorescent assay to actually assess the damage of your double-stranded breaks from this reactive oxygen species. And so you could have a mitochondria with a lot of copy number and it's functioning really well and it's really healthy and has a high efficiency, whereas you could have a lot of mitochondrial DNA that is damaged and this mitochondria is not doing so well. So it's another kind of thing, another piece of the puzzle of how these things are all interacting with each other. So with that, I would like to thank you guys for listening, and if you guys have any questions.
Great, Holland. Thank you so much for the great talk connecting mitochondria and telomeres, something I had not thought of before. So we will start off with the questions. And Sarah Scott has her hand raised, and I will allow you to talk, Sarah. But you may have to unmute yourself. Oh, it was accidental. Okay. So I will, well, I'll disable talking then. I feel like I have so much power here. Yeah, I'll start off with questions while we're waiting. I was wondering, did you monitor multiple chicks within the same nest, and how did you control for that effect?
So for the metabolic rate, it was only one chick per nest that they
Okay.
took. So we did not do multiple chicks across the nest because it would be interesting to look at that because brood size affects it, which I need to go back and rerun the model with growth rate. At least we know brood size affects it. Also, we know that your hatch order affects it too. We know earlier chicks that hatch are usually larger than the last chicks who are smaller. So again, that body size connection could be, connected as well based on your hatch order.
Yes, and so along those lines, the dates where they're nesting are very variable across the year. Did you
Hmm.
look, were there any multiple clutches, like the same parents across the year, or was it always different?
But that's a good question. So this was trying to think back how this was all collected.
Hmm.
So I was during this time collecting adults, and I could go back and actually probably look at if there was repeat adults for that because house sparrows do usually probably have three broods approximately per individual. I don't necessarily know if they stay with the same pairs, but that is a potential source that we could look at. And I do have the data. We caught like over 100 adults during that time. And I think some of them do correlate with the nest that we did metabolic rates on that. Because that could be interesting because we also have feeding rate videos too and incubation videos. So we can actually maybe match more of the behavior and how that might be affecting if some are compensating for cooled nests versus controlled nests or stress nests versus not stress nests.
Yeah, I'm just thinking about the genetic component to metabolic rate and telomere length, and if it's the same parents that are contributing to multiple data points, whereas other data points have a single
Mm-hmm.
parent contributing, that could be a potential factor in the analysis, and
Right,
it could change things to be significant after it's
right,
controlled. Yeah.
right. Yeah, like we could run because at least have run the copy number on the males from that time period. So we could look at how those. That's the question. Is I don't know how heritable copy number is from parents, either, as well as telomere lengths. I know we have looked at. I think that's a tentative plan is to look at how telomere length influences chick telomere length. I think that is a project that is ongoing in our lab as well. So,
Got you. Yeah. Clutch size was my other comment I was going to
mhm.
make. Um, I was just wondering, I apologize if I missed this, but what was the stress that the chick went under?
So the stress was just literally physically removing the chick from the nest for an hour a
Mhm.
day. We put it like in a little bag, bird bag, and then put it back to mimic like parental neglect. So they're not getting fed as much, quote unquote. It's kind of that stressful condition. Also, the stress of handling them as well.
Ah, got you, got you. And since it was one, I had been thinking it was multiple birds in a nest. Since it was one bird in a nest, did you pick so it did vary across if it was the firstborn
Hmm.
or
Yeah.
whichever. So,
So what we do is funny. We actually color our chicks with Sharpies so we can tell who it is throughout the whole time that we're measuring them. So what we did was actually just randomize it with like a die, and like it was like even was like stressed, and odds were cool or control. So that's kind of what we did for randomizing it, and then we would pick one from those nests how for metabolic regimens because we didn't want to take too many chicks from the nest at that same time for potential abandonment for our parents as well.
got you. But
But we
if
have
you, yeah, no, go ahead.
I was gonna say we have a paper published that was looking at how cooling and stress affects across the whole nest as well, not just the single individuals that were metabolic rate have been taken
Okay.
on. So
So I probably need to take the time to read that. I should go check it out.
yeah.
So should everybody else here. Um, but so if you take do so you have a record of if they were the first or how big is a clutch? I'm sorry.
Five. Usually about five is what they go around. Some could have a little bit less. I really haven't seen many that have laid more, but my memory could be foggy.
So
Um,
if you
sorry,
yeah, if you take
what was your question?
sorry, that was one question. But then like if you control for like just firstborn or just fifthborn or whichever number, and then look at the relationship, since it would
Hmm.
appear that clutch size or birth order does affect it.
Hmm.
It would be interesting if those variables are clouding some significance. Yeah.
Absolutely, absolutely. I think we can potentially do that because like what we do for hatch order in our population, since we do color them, we usually assume the bigger one is the firstborn, right? And then the smaller one is the last. And
Mm.
so that's kind of like our proxy of it. So I know there was some studies that we tried to like actually mark the eggs and then see which ones hatched in order. I don't know how successful that was in other field seasons that we've done here. But yes, we could definitely put that into the models as well to see how that is affecting it.
Uh,
Actually gonna
yeah.
write that down.
Yeah. I just remember. Or yeah, I was just interested to see if uh, tease apart some of these um, results that didn't turn out significant.
Hmm.
Um,
Yeah,
and then I just had methods. Actually, let me just make sure. I'm sorry. Uh,
that's okay.
Carolyn asks in the Q&A, did you notice any parasites on the nestlings or in the nests? Yeah, great question. Did your study include multiple clutches from the same nests or parents, which is what I asked earlier?
Yeah, so our study did, that's when we have to go back and verify if the clutches were from the same parents, if we caught parents at the nest, or if we were able to video ID them. Um, Parasites, I know there are parasites. I don't know if we took notes of them back then. I'm not quite sure. So that could influence it. I know like when we work with our great tits in Germany, we do take note of parasites on chicks, but I'm not quite sure if that was done. But that could definitely influence it. You know, if you have more stress with parasites, that could increase more stress and metabolic rate or have some kind of other physiological change or shortening of telomeres. But that's a good point as well.
Exactly. And Sarah Scott does ask, thank you for this great talk, Holland. Is there much variation between sites in nest density and or aggression between adult pairs? Curious how this might play into stress levels in adults and nestlings.
Yeah, so our field site is composed of like four buildings across this farm area. It's like an NDSU USDA farm that they're allowing us to use. Nest boxes are a space, I think like about God, 8 feet apart. So there's high density
Hmm.
between them. And we have seen aggression and nest takeover in these boxes as well. We actually had a little serial killer in 2023. Some rogue male was going out and killing a whole bunch of nestlings, actually. So yes, that could have taken effect. Because it's a very dense population. There's like about 200 nest boxes all spaced within that as well. I'm not sure how we would incorporate that. That could be more of a parental behavior videos we could potentially look at as well. We have hundreds of hours of behavioral data that we could go through and kind of parse that out.
So yes, opportunities for future graduate students as well, I'm
Yes,
sure.
yes. We have a PhD student right now. She's working on parental behavior for feeding and incubation for a lot of these videos.
Cool. Well, guys, we are at 1 o'clock over here on in the east, and so we are gonna wrap it up. But thank you so much, Holland, for the great talk, and I certainly learned a lot. And just thank everyone for being here today.
Thanks again for everyone listening. It was wonderful to give a talk with you guys.
Bye, guys.